we have finished. Okay, life cycle in human. Life cycle in mosquito, if you see here in detail, I will make it down only. Okay, separately I will make it below. Right. Then we can later incorporate. I'll incorporate in the notes, and I will see that if it is possible in one page. I don't think so. It is possible, but if possible in one page, a simple one I can give you later. Okay. You can go with the NCRT thing also, not a problem. NCRT in a very simple and crisp way they have given.
Sir, once could you scroll up and zoom out, sir?
This was done earlier. This zooming out thing was done earlier. Please let us finish now. If we go on for that only, it will take more time.
Okay.
We can do that later. I said now I will be sending it. Don't worry. Okay. So life cycle of Plasmodium. Life cycle of Plasmodium in mosquito. Which mosquito? Tell me. Which mosquito is involved?
Female Anopheles mosquito.
Female Anopheles mosquito.
Female Anopheles. Female Anopheles mosquito, right? Okay. So what happens is we will start from bottom because again we will send it into the human, right? It will the same thing we can copy and in a small area we can paste it over there. Now let us say the mosquito. Okay, in the mosquito, who are coming inside the mosquito body? Tell me. Gametocytes are coming in, right?
Yes, sir.
Yes, sir.
Okay. So along with blood meal, mosquito is taking blood meal. Healthy mosquito is going and drinking blood from an infected person. Okay. So blood meal contains, let us say these are the RBCs. Okay. And RBCs are having what? Gametocyte. Okay. This let us label it as a gametocyte. Okay, RBC with gametocyte. Now, along with blood meal, it will enter. So if a mosquito drinks blood, where will this blood go? Tell me. It will go into the stomach, or we can say into the alimentary canal only, right? Okay. So now, what happens in the alimentary canal? In alimentary canal, or in a very simple word, I just write it as gut. In a gut. Okay. What will happen? RBCs are digested. Keep your mics on mute. RBCs are digested. And gametocytes are released. Okay. And gametocytes are released. Okay. So here what I will do is, I'll make this as a microgametocyte, megagametocyte. Okay, so this I write it as a microgametocyte, and this as megagametocyte. Okay. Now this microgametocyte, okay, very, very important here is this microgametocyte, it undergoes changes. It will undergo small change. Okay, it will undergo change. And that change is called as exflagellation. It undergoes exflagellation. As a result, what happens is the nucleus is there, no? The nucleus will divide into 6 to 8 in this way. 8 I am drawing. And the cell also will divide in this. The point I will give you later when we give in the form of points. In this way, exflagellation occurs. As a result, 6 to 8 nuclei will be divided. Okay, and around which the cytoplasm is collected, and they will form long flagellated structures. So in this way, flagellated structures. This act as what? These act as microgametes. Now this form microgametes. So the microgamete will be formed. Microgamete is nothing but what? In a simple way, we can say it is the sperm. Okay. And what about the female? That is the megagametocyte. Megagametocyte undergoes division. But this division is unequal. Okay. The nucleus will undergo division. Okay. The division is unequal. Right. So one will be large cell, one will be smaller cell. It will act as a polar body, and this polar body will degenerate, and this forms the megagamete. This forming the megagamete. Megagamete is nothing but just like a egg, right? Is it clear? Now, this megagamete, once it is formed, this megagamete, it forms a projection, okay, small projection in this way. Projection is formed. This projection is called as cone of reception or cone of fertilization. Okay. Now, what will happen is the sperm and ovum, the sperm will fuse with ovum. So this process, what do we call? Fusion of sperm and ovum?
Fertilization.
Fertilization. Okay. Once fertilization occurs, there will be development of a zygote. This is now
Yes, sir.
zygote. Okay. Now, what will be the number? What will be the chromosome number in zygote? Tell me. It will be doubled, right?
Yes,
Yes, sir. Diploid.
Here I can say this is haploid N. Haploid. This is also N. Microgametocyte is haploid. This is also haploid megagametocyte. So gametocyte itself, we can say it is a haploid structure, right? Okay. Haploid. Right? And now, once the zygote is formed, two haploid, when they combine together, it will give rise to diploid structure. So zygote becomes now 2N. Okay. Now this zygote, it will develop a projection. This zygote, it develops a small projection in this way. Okay. And it becomes motile. Okay. It is called as ookinete. Ookinete. Ookinete is motile zygote. Now it will come out of the gut wall. Okay. Remember we had the gut wall. 1 second.
Yes,
I can just what I will do is the darker line.
So what happens to the polar body?
Polar body will degenerate.
Okay.
Okay. Let us say this is the gut wall. Okay. So here we say this is the lumen of gut. Okay. And this is the gut wall. Okay. And this is the body cavity or coelom, right?
Sure.
In arthropod, coelom will be there, right? Now what happens is, now this ookinete, it will try to come out of the gut wall. Okay. So to come out of the gut wall, what it has to do? It has to make one hole, right?
Yes, sir.
It bores out of the gut wall, now like this, out, making a hole, and it will come out of the gut wall. Okay. And then after coming out, it will get settled on the outer wall. This spookinate, it settles on the outer wall. Now slowly, once it settles, okay, the okinate as well as the gut wall, they secrete a layer around this okinate. Okay, this okinate is there, right? Now around this okinate, a layer will be formed. A covering, protective covering will be formed around it. And this covering is formed by whom? By the okinate itself and also by the wall of gut. Okay, so now this is called as what? This is called as formation of cyst will be seen. Still here, if I take the ploidy, this is 2N diploid. Okay, now within the cyst, inside the cyst will also grow. That growth will be seen in the cyst. Within the cyst, the first division will give rise to 2 cells. Okay, so here, first division is meiosis. What type of cells will you obtain? Tell me. Haploid cells. So say here, from here only haploid cells begin. Okay, and after this, multiple fission will occur.
Sir, 4 cells, no sir.
Wait, na. First, in cell division, first cell division is meiosis, right?
Yes, sir. Yes.
After that, there will be mitosis only, na.
Yes.
Second meiotic division is mitosis only.
Yes.
So after that, later on, there will be multiple mitotic divisions. Continuously, the cell will undergo division by mitosis. Okay, continuously it will start dividing by mitosis. So next divisions are mitosis. Next, all the divisions are mitosis. Okay, and the cells, the resultant cells, these are sporocysts. Okay, these are called as sporocysts. Okay, so here the whole structure we can call it as sporon. Okay, the sporon, the whole structure of sporocyst, and when they come out slowly, it will break, and these structures, they come out and enter what? If they come out, they enter into the body cavity or the blood of the cockroach, of the mosquito, right?
Yes, sir.
What are these structures that are coming out? These are sporozoids. Sporozoids are released, okay, by rupturing the wall. Okay, by rupturing the wall of cyst, the sporozoids come. They enter into the hemolymph, and from hemolymph, they enter into the salivary gland. Within the salivary gland now, if you say, within the salivary gland, okay, sporozoid, sporozoids are stored. Sporozoids are stored. Now say, when this mosquito who is having sporozoid stored in its salivary gland, okay, if this mosquito, it will bite a healthy human, then what will happen? See, for drinking blood, what will the mosquito do? It will inject its saliva, right?
Yes, sir.
In order to prevent the clotting of the blood. So at that time, from this mosquito, okay, along with saliva, here I can just write, along with saliva, sporozoid enter human. sporozoite will enter human. This is how it will complete its life cycle in mosquito. Okay, then come back to again humans. Got the point? Any doubts with respect to mosquito? Any doubts with respect to mosquito?
Sir,
Nothing? Tell.
generally after meiosis we will form 4 haploid cells now, sir. Here you give only 2 haploid.
Here what did we say? First division. After first meiosis, let us say in meiosis, do you remember in meiosis?
Meiosis
two cells are formed.
Meiosis one.
Mitosis.
Meiosis 1.
So this is meiosis 1 where there will be from 2n to n.
Yes, sir.
Then this will divide again
Into meiosis 2.
So here also it will be n only. This is meiosis 2, which is equal to mitosis. Here what did we say? Next division are mitosis.
Yes.
Okay, sir.
And after this mitosis division is continuing. That is the reason many cells are formed. Already one person spoke about it and we explained, even though you want to hear again and again. Not to see many cells we have made. Step by step we are taking, right? Many cells are there, not one or two. I hope now you got the point.
Yes, sir.
Okay. See, one more just thing I'll give you. See, the division over here is also asexual division only, right? After the cyst was formed, after the zygote was formed, zygote is dividing by
Fission.
asexual mode of division, multiple fission only, right? So this is called as sporogony. Remember we said sporogony will occur in case of mosquito.
Yes, sir.
Yes, sir.
Asexually formation of sporozoite. Okay, and gamete formation and gametic fusion. This is the sexual stage, right? Sexual stage, we can call it as gamogony. Gametes are formed and gametes are fused. Any doubts still here?
No, sir.
No? So now what I will do is in the form of certain points. Let us see. Okay. So that it becomes little bit convenient for you.
So it's a one scleral.
I'll do it. Which part is required? Tell me.
Done, sir.
Is it done?
Yes, sir.
Okay. So pointers if we take. Shall we start with human then?
Yes, sir.
Yes.
So listen very carefully and take point by point on. Okay. So sporozoites are infective stage. For human, infective stage for humans. Okay, and remember, sporozoites entered the human blood. From mosquito during blood meal. Okay, for this you must remember, mosquito injects saliva containing sporozoites. So mosquito is not deliberately pumping the sporozoites. See, mosquito does not want to send sporozoite. Mosquito wants to just send saliva. Okay, so saliva for what? To prevent blood clot. And its saliva will have anti-clot proteins. But in saliva, sporozoites were present. As a result, sporozoite enters. So sporozoite is for haploid stage only, right? Isn't it? So this is how the infection in human occurs. Clear about the first thing about infection? After infection, what will happen? Within 30 minutes of inoculation, the pathogens or the sporozoites disappear from blood and enter the parenchymatous. Parenchymatous cells of liver. Okay, they are hepatocytes. Okay, called hepatocytes. Now, in hepatocytes,
Sir, inoculation.
inoculation, inoculation, means entry. So in hepatocytes, sporozoites reproduce asexually, and what is this called? Tell me. Schizogony, right?
Yes, sir.
Schizogony, and release merozoite. Okay. First generation merozoites are called what? Are called cryptozoites. Okay, the first generation are called as cryptozoites. Are called cryptozoites. These merozoites re-enter merozoites or cryptozoites reinfect new liver cells and undergo schizogony and release second generation merozoites called metacryptozoites. Right? Now, these merozoites, if you see, okay, these merozoites, okay, will be released in the blood. Now here you can see. The merozoites, after few cycles, are released into bloodstream. And tell me, what is the time gap between the entry of pathogen and its first appearance into the bloodstream called?
48 hours.
Pre-patent period.
8 days.
Pre-patent period. Okay, so time gap between entry and first appearance of pathogen in blood. Okay, so that is pre-patent period. It is 8 days in Plasmodium vivax. Okay, now the merozoites, here one point I can say that merozoites that infect the RBCs are called as phanerozoite. Okay, these are called as phanerozoite, these are nothing but phanerozoites are what? Phanerozoites are metacryptozoites only. Okay, so here we can call it as metacryptomerozoites. See, in order to make it little simple, which is entering where? See, first generation cryptozoite, second generation metacryptozoite. Okay, and the one that will not enter the liver, rather it will go and infect the blood, metacryptomerozoite. Okay, or phanerozoite. Okay, and within RBCs, in RBCs, the merozoites divide asexually. So over here also we can say this is called as erythrocytic. Erythrocytic schizogony. Okay, erythrocytic schizogony. Here we can say it is pre-erythrocytic. The first one was pre-erythrocytic. Now this is erythrocytic schizogony, also called as Golgi cycle. Okay, also called as the Golgi cycle or cycle of Golgi. Okay, so in this case, what is happening? See, in RBCs, within RBCs, so merozoite develops into trophozoite. And will be which stage? Amoeboid stage is received, right? First signet ring, right? Signet ring stage, then amoeboid. Signet ring stage. Okay, then amoeboid. Then rosette stage. See, when amoeba starts dividing, amoeboid stage starts dividing, it will look like a flower. Okay, that is the reason it is called as the rosette stage. And as a result, from here, after the rosette stage, okay, what is happening? The merozoites will be released. Okay, so merozoites, many merozoites will be released. How many? Around 12 to 24 merozoites are released. Okay, and these merozoites, the merozoites released from the RBCs, released from RBC are called schizozoites. Okay, so with the release of pathogen, hemozoin is also released in blood. Hemozoin is also released in the blood. Okay, and this hemozoin causes onset of fever with chills. So this is the one that causes the disease. Causes onset of fever with the chills. Right? Everything was explained already. And now tell me, who is the infective stage for, who is the pathogenic stage for man? We can say the merozoites, right?
Yes, sir.
In liver also they are causing damage, right? All are merozoites only. Merozoites, pathogenic stage for man. Okay, it causes damage, right? And merozoites will cause damage to hepatocytes. Okay, as well as damage of RBCs, right? Because they'll rupture the RBC and damage RBC and feed upon hemoglobin, As a result, Schaffner's dot, do you remember Schaffner's dots?
Yes, sir.
Yes, sir.
Schaffner's dots are formed. Right? Now, some merozoites, okay, some merozoites within RBC do not divide and become large. Some merozoites in RBC do not divide and form gametocytes. Okay, can give rise to gametes. Okay, but say the condition is, but for gamete formation, lower temperature is required. And tell me, human, is it a warm-blooded animal or a cold-blooded animal? Warm-blooded.
Warm.
So can this gamete formation occur in human?
No, sir. No, sir.
No, na. So remember, but as humans are homeotherms, okay, gamete formation of Plasmodium is not favored. Okay, so these gametocytes, once they are formed, if they do not enter into the mosquito, they will die in the human. Okay, if they don't enter into the mosquito, they will die into the human only. Got the point? Okay.
Yes, sir.
Now, along with blood meal,
Gametocytes enter the mosquito.
Gametocytes enter the gut of female Anopheles mosquito. Okay, so see, they can enter any mosquito. Can enter any mosquito that bites, but development will occur only in female Anopheles. Okay.
Sir.
So certain things that are required for its development, they are present only in female Anopheles, the proteins, enzymes, and all. Okay. Now in the gut, RBCs are digested and gametocytes are released. And what are the two types of gametocytes? Tell me. Gametocytes were two types, right?
Micro and mega.
Microgametocyte and megagametocyte. And megagametocyte. Okay, this microgametocyte undergoes exflagellation. Exflagellation. Okay, and forms how many? It forms 6 to 8 microgametes are formed. Okay, and these microgametes are flagellated. Okay, so nothing but we can say in this way, sperm type, right? Elongated sperm. Okay, and megagametocyte, if you see, undergoes unequal division. Okay, as a result, forms an egg and one polar body that degenerates.
Sir, sir, what you wrote below microgametocyte?
Form microgametocyte undergoes exflagellation.
Okay, sir.
Okay.
Sir,
Now
sir,
here-
what is after polar body that
Polar body that degenerates. Degenerate.
okay, sir.
Now the egg forms a cone of fertilization. Cone of fertilization. Right? Next point, if you see after the formation of gametes, what will happen? Gametes fuse to form zygote in a gut. Okay. Zygote develops an extinction and becomes motile okay, and is called what? Just now we saw right, ookinete, and it forms ookinete, right? And what will ookinete do? Tell me. Ookinete bores through.
Sir, it moves out of the gut wall.
Ookinete bores. Array, switch off your mic. Bores through gut wall and settles on outer gut wall in coelom. Okay. Next, it forms ookinete and wall of gut form a cyst around the ookinete. Okay, now it will be called as what? Sporocyst. Right? Within the cyst,
Sir, can you show the second point of mega?
array, after completion of so many points, now you want to see the second point of megagenesis, right?
Then scroll
That is slow. Within cyst, it divides.
In that, what is that word, Serpida or something?
It's not a PEDA. That is the reason you have to listen also carefully. Not listening, you were somewhere, now suddenly you came and you're asking polar body. After explanation, when I give the points, even then you have problem in understanding things. God only can help you. Within cyst, it divides first by meiosis and then by mitosis. Okay, and forms many sporozoites. Okay, rupture of cyst wall. Releases sporozoite into the hemolymph, which reach the salivary glands and are stored in it. Okay. These sporozoites infect a healthy human during blood meal. So we started with blood meal with some human and we ended at the blood meal. This is how the whole life cycle of Plasmodium is taking place. Is it clear? Is it clear?
Yes, sir.
Already explanation was done.
Yes, sir.
Yes, sir. Can you go?
Sir, last is
Sir, last. Sir,
So the emotions, sir.
sir last point.
Last point, the sporozoites infect a healthy human during blood meal.
Emotion line, sir. Emotion.
Hemozoin line. Because of redoing it, doing it again for years.
Hemozoin is like a toxin, sir.
It is a toxin. Already we said, it is a toxin.
So released by the merozoites.
It's not released by the merozoites. In the RBCs only it is formed because of merozoites.
Okay, sir.
Got the point? Is it
Yes,
clear?
sir. Yes, sir.
Sir, could you please
Only 2
last
points.
2 points?
Only 2 points more, like a treatment. For treatment of malaria. Okay, there is one drug, quinine. Or quinone also. Okay, it is a drug obtained from bark of cinchona. It is obtained from the bark of cinchona. It is a common drug of choice. Normal antibiotics can also be used, but this is a favorite drug of choice. Okay. And prevention, how you can prevent, tell me? Prevention by preventing the
Sanitation.
Stagnant water.
growth of mosquitoes in water. Okay. And you would have learned in your lower classes, Gambusia.
Yes, sir. Left in
Sir,
the
yesterday asked sir that question.
Yes.
Gambusia, it is a larvicidal fish. Okay, means eats larvae of mosquito. So even this will kill, right? It will prevent the mosquito growth.
Yes.
Okay. Then insecticides could be used. Generally, DDT is used, right? Okay, DDT spray and all they will do, right? So this is all about malaria. Next, we will start with the helminthic diseases. Just I'll give helminthic diseases and then we can end it here. Okay. Some common helminthic diseases.
Sir, DDT.
DDT is an insecticide. I hope you got the botany link also, right? You can go and join the botany class.
Thank you, sir. Bye.